The following program is brought to you by Caltech.
The first session was extremely fascinating, and as a neuroscientist, I'm very much looking forward to our next presentation, Ralph Adolphs. So our second presenter received his bachelor's degree from Stanford University and his PhD in neurobiology from Caltech, so a fellow Caltech alum. He did postdoctoral work with Antonio Damasio at the University of Iowa, beginning his studies in human neurophysiology with a focus on the recognition of emotional facial expressions. Since 2004, he holds an appointment as professor at Caltech, as well as an adjunct appointment in the Department of Neurology at the University of Iowa. He is the director of the Conte Center, the Neurobiology of Social Decision-Making, the principal investigator in Project 4, Connectivity of the Social Decision-Making System, and the principal investigator for the administrative Core 1. His research has covered many different topics in neuroscience, psychology, and biology, including decision-making, social behavior, vision, neurophysiology, and brain connectivity, and psychophysics. Ladies and gentlemen, I am pleased to introduce the Bren Professor of Psychology and Neuroscience and Professor of Biology, Director of the Caltech Brain Imaging Center, Dr. Ralph Adolphs.
Thank you very much for that kind introduction, Heather, and thanks to you and everybody for organizing this event, in particular Phil Scanlon, who's put in a huge amount of work, I know from personally interacting with him over the past several weeks. And thanks to all of you for coming here. Can you all hear me well with this lapel mic? Okay, great. So my lab works on a lot of things, as Heather mentioned, but what ties them all together is the quest to understand human social behavior. And so I'll be telling you some basic science about how we think that works in the human brain, and studies of one particular clinical population where it breaks down, which is autism. And let me start by just giving you an overview of what we'll be doing in the next hour or so. I'm going to start with with a background and introduction to autism, say a few words about the social brain, and then move on to one story, the earliest story really, where we tied together people's motivation or attention towards social stimuli, or propensity to look at other people's faces in particular, and one particular part of the brain, the amygdala, that plays a critical role in that. Then tell you a little bit about recordings at a much finer level of analysis from single cells that we've been doing in collaboration with some hospitals nearby. And then extend this out to the real world and ask the question, how can we begin to translate findings in the lab to actual social interactions in the real world, which are, of course, much more dependent on context, much more complicated. And then also zoom out to talk about brain structures other than the amygdala and ask how the whole brain, how the social brain subserves this. We'll have Q&A after this, and then I'll spend sort of 5 or 10 minutes right before 12:30 to give you an introduction to the Caltech Brain Imaging Center, just a quick overview, since that's where many or most of you will be touring next. So by way of background, there are many different levels of analysis at which we want to understand social functioning, behavior, and so forth. Clinically, and in terms of what you observe in the real world, what's sort of most relevant is the final one over here, how you actually function in the world, how you interact with other people, how you're able to make and maintain friendships, how you're able to hold a job. We live in such a social environment that essentially everything that you do that dictates your career success depends on social interactions. And of course, this depends not just on you, but also on the other people that respond to you and that interact with you. So it's a very complex domain to study, but it's the one that, for instance, for clinical populations is the most relevant. We're interested in, say, how people with autism function generally in the world. That, of course, depends proximally on actual social behavior, and that's something that we can begin to quantify more in the lab. So we can, for instance, have dyadic interactions between just two people, measure how they make eye contact, and try and quantify how people interact in more constrained settings than the real world. And of course, what makes this possible are various psychological processes: perceiving faces, remembering names, decision-making, motivation, attention towards social stimuli. And then ultimately, all of that has to be implemented in the brain. and in particular parts of the brain, collectively called the social brain, that mediate all of this. So these are the different levels of analysis, and of course you can, within the brain, go down to single cells, genes, and so forth. And the challenge is to try and tie these all together to tell a story that ultimately explains how genes and environment form neurons in the brain that interact in ways that give rise to social behavior and social functioning, which is not to say that these higher levels are reducible to the lower ones, but they certainly depend on them in a strong way. So the social brain, shown here, is a collection of brain regions, shown in all these different colors, that mediate our social behavior. We don't know what all these regions are yet, so this is a very active research endeavor, informed mostly by functional MRI imaging, some lesion studies, some studies in animal models. And the important take-home message is that there are many regions of the brain that are important for social cognition and social behavior. And these regions are all interconnected. So asking the question, what does this one purple region down here do? While we can get some traction on that experimentally, isn't really the right question, because in isolation, if you cut it off from the rest of the brain, it doesn't do anything at all. So we need to understand what it does in terms of where it receives its inputs from, where it projects its outputs to, and how it functions as one component of a neural circuit. So all these different color regions are connected, and you should think of them as a system, but we can still ask questions about what it is that each component in a system contributes to the functioning of the whole. And we'll be talking in particular about this orange region here, the amygdala, which is one brain region that my lab has studied in great detail. So to give you the conclusions of what I will be telling you about the social brain and the relation of that to autism, I just want to preview what I'll be telling you. So I'm going to be telling you that social cognition depends on particular brain structures like the amygdala that interact, they're connected, they're components of a distributed network which we can measure with a variety of different techniques. And so they have to be connected. There has to be wiring that connects these brain regions, white matter tracts that connect these distal brain regions. I'll be telling you near the end of my talk about one population in which the largest white matter tract in the human brain, the corpus callosum, which connects the left and the right cerebral hemispheres, is missing. These are people with agenesis of the corpus callosum. And then a lot of these connections seem to have gone awry in people like autism, in part explaining the difficulties that they have in social functioning. And they also show large individual differences in healthy people. So it's important to note that autism spectrum disorders, is a spectrum, and there are many differences also in typically developed people that depend on how these different brain regions are connected. So let me turn to autism, which has been extremely visible in the news, and the covers of what you see here already make some points about autism. It's a pervasive developmental disorder, so it's diagnosed around age 3 or so. Most of the research going on, most of the attention has been focused on infants and children, but once you're diagnosed with autism, you have it for the rest of your life. So of course, all of these children and infants will age and grow up to be adults, and they will then age, and actually very little is known about autism in adults and how autism and aging interacts. Now, the other point to be made here is that autism is highly heritable, so it depends on genes to a very large extent, and part of that is reflected in the gender bias in autism. It's much more common in males than in females by about a ratio of 4:1, and hence all these covers are males. So just to summarize some points, the current prevalence is thought to be about 1% for autism, which is remarkably high and higher than it was thought to be at various points in the past. This graph on the left here plots the diagnosis of autism over the years, and you can see it's just shot up. And one question, of course, is why is that? And there are probably two factors explaining this. One is that it's just diagnosed more readily. Children with autism come to attention more readily. And the second one is perhaps there's a real biological contribution to this as well. So this pie chart over here on the right summarizes what we know that might explain this increase in the diagnosis of autism over the years. And you'll notice that mostly we don't know what causes this, which is why there's the big question mark here. But to some extent, it's that people just are diagnosed more readily. There's more awareness of autism. There are some biological factors, such as that parents tend to be older nowadays than they were in the 1970s, and there's a higher risk of having a child with autism if you're older than if you're younger as a parent. In terms of the consequences, both for the individuals, for their families, and for society, neuropsychiatric disorders collectively are a huge impact. This is a rather strange unit here. This is disability-adjusted life years, which sort of combines mortality and morbidity together. But what it tries to map is the number of years of healthy functioning that you would have. So if you die prematurely because of cancer or a heart attack, that would obviously truncate the years that you would have. But you could also have years of disability while you're still alive, and that's factored into this metric. And if you use that metric, neuropsychiatric disorders, schizophrenia, mood disorders, autism, collectively account for a huge proportion here. They have a much greater burden, both socially and economically, than does cardiovascular disease, cancer, and many other really, really big things. So it's a major problem, the major problem in terms of public health to turn our attention to. And certainly in the case of autism, we know very little about what to do about it. So what is autism? It's diagnosed by this thing up here, which is the Diagnostic and Statistical Manual of the American Psychiatric Association, DSM. Edition 4, until this last May, there's now a new edition out, edition 5, which has some differences and, for instance, makes no longer distinctions between autism and Asperger's syndrome, but pools them together under autism spectrum disorders. Nonetheless, most people currently still use, and most of the research and the published papers are based on criteria from DSM-4, which has these three. So it diagnoses autism spectrum disorders by dysfunction in these three domains: social impairments in terms of how these people interact with other people, communication deficits, often involving delayed language onset, and very narrow, often stereotyped interests and behaviors, not for social stimuli, but for geometric patterns or for mechanical gadgets and so forth. What we want to do ultimately, and what the research has focused on, is to understand autism at all of these levels. In terms of distal causes, there are genetic and environmental factors that interact in complex ways that give rise to changes in how the brain is connected up, that give rise to changes that we can measure in terms of how the brain functions, that ultimately give rise to changes in behavior on the basis of which autism is diagnosed. And so we want to link together all these different levels of analysis, not just have a slide or two here on the genetics, which is a topic to which a huge amount of research effort is allocated, but one that my lab doesn't work on at all. We're at the other end of the scale in my lab. But as I mentioned, autism is highly heritable. So if you have an identical twin who has autism, the chances of you also having autism are greater than 50-50, up to 90% by some estimates. And if you have a non-identical twin, you also have some chance of having autism. As I mentioned, it's much more common in males than in females. This ratio is pretty closer to 10-1 or even more when you look at high-functioning individuals with autism. Despite the fact that it's highly heritable, and so there are many genes involved that predispose you towards a risk of developing autism, there are hardly any single genes that can really predict you developing autism. So this shows this here. What's plotted here is the influence that each gene has individually in the y-axis, and then how frequent these particular mutations or copy number variations in these genes are. So there's a lot of common genes, but each of them has a very, very small effect. So collectively, if you had many of these particular alleles, they would add up to a substantial risk for developing autism. But individually, they do very little. There are a few very rare ones, like genes for tuberous sclerosis or Fragile X and so forth, that do have a big effect size. And in those diseases, you have a high proportion of people developing autism. So there's a strong genetic predisposition, but it's not because of a small set of genes. It's spread out over small effects over many hundreds, possibly even thousands of genes. How this then leads to the phenotype, to autism, isn't known, but the presumption is that it depends on a complex interaction between all these boxes shown here. So you have a genetic predisposition due to the action of many different genes. You have environmental factors that also make a contribution, and then this gives rise to changes in the way that the brain develops. Also very important interactions between brain development and immune function that eventually give rise to an atypical connectivity in the brain that gives rise to the cognition and behavior that's diagnosed as autism. So what have we been looking at in this tremendously complex network of causes and effects that give rise to autism? We've been looking at one particular process that is thought to be critical, especially early on in life, in explaining why many people with autism in particular have difficulties in social functioning. And this is early disruption in the motivation or attention towards social stimuli in the first place. So you could imagine if you have an infant and this infant is not motivated to look at or pay attention to the mother's face, the infant's brain isn't going to be getting normal visual input about faces and isn't going to develop, therefore, the specializations for face processing that you and I have. So the idea is that early on in life, if you have even a slight imbalance in the propensity to be motivated by, to find rewarding, to pay attention towards other people, that this would give rise to reduced social input developmentally, which in turn gives rise to reduced cognitive and neural specializations in the brain for processing information about people. And then this is kind of a positive feedback. If you have this, you have reduced social input and so forth. So you could imagine early on, if an infant isn't motivated to respond to his or her name, to attend to faces normally, that this would translate to an altered developmental trajectory along which the child develops that ultimately translates into autism. And so we've been looking at this process in relation to faces and social attention, and in relation to one particular brain structure where we think we understand these processes the best, which is the amygdala. Let me just show you a quick video that makes the point that faces are extremely salient to all primates. This is a newborn macaque monkey. They're much more mature at birth than our humans. And what you'll see here is this baby monkey is fascinated by the experimenter's face. So the face is an extremely attention-grabbing stimulus, so much so that the little guy's just watching this face. And you can imagine, as he's doing this, his brain is processing information about the features of this face. And if you do this a lot, your brain is going to be tuned up to processing faces, to distinguishing the faces of different people, to associating certain facial expressions with certain emotions. All these are things that you need to learn early in life. But you need to have a mechanism in place that makes you learn this in the first place, that makes you find faces interesting and look at them and pick up that information. We can quantify this in humans. We don't use head-mounted eye trackers in my lab these days anymore, but it makes a clearer point. We can measure with these little video cameras down here where someone's direction of gaze is as they look at a face. And so we can quantify where people look in the world. And if you're looking at a photograph of a face, you look at it more or less like is shown in this little video here with that spotlight moving. So there are two important points to make about this. One is that you explore the world with your eyes and you explore faces with your eyes. It's not like taking a snapshot with a camera where you just open your eyes and the information comes in. You have to look and seek out social information to pick up in the first place. And you look preferentially at different parts of the face. So you're not so much interested in looking at the chin or the top of the head. You spend most of your time looking at those regions of the face that carry the most information, which are the eyes and the mouth region of the face. And so in my lab, what we've been doing is asking the question about one particular aspect of faces, which is emotional facial expression, and asking the question, how does your brain figure out from these various configurations of faces what the person might be feeling? And there's been many studies of this, including cross-cultural ones, making the point that across many different cultures, people reliably recognize these 6 so-called basic emotions from facial expressions: happiness, surprise, fear, anger, disgust, and sadness. And people have developed techniques, which I'll show you one of in just a second, to figure out what are the parts of these faces that carry information. Which parts do you attend to in order to figure out what the emotion is? And for happiness, it's the mouth. For surprise, it's a big open mouth. For fear, it's wide eyes, and so forth. So a lot of these make some intuitive sense. There's sort of a vocabulary of regions of the face that change, that transmit information about the emotion that the person is feeling. So to turn to our studies, we've studied in great deal this particular structure right here shown in orange. So if you take a view of the brain from the side, like is shown here, and you were to cut it down here, then down here, sort of just going in from your ears, is this structure, the amygdala, in the medial temporal lobe. And we've been studying this structure for a number of reasons. One is that we have rare patients who have damage just to the amygdala, but nowhere else in the brain. And so we can ask, what is it that this component contributes to the social brain and to social cognition and behavior? And there's a lot known about the function of the amygdala. In particular, it's known that it gets inputs from sensory cortex, from visual cortex, that would tell it there's a face out there in the world. And the amygdala then can modulate many other cognitive processes, emotional responses, encoding into memory, attention, decision-making, on the basis of the value or the emotion that's shown in the face. So the experiment that we've been doing, a set of experiments we've been doing, revolve around very rare patients who, because of a genetic disorder, have bilateral lesions of the amygdala. This is one particular patient whom we've studied the most, patient SM, and you can probably appreciate her lesions best in this horizontal cut A up here. So where these dark circular regions are is where her amygdala should be, but it's lesioned in her case. So an extremely specific brain lesion. You would never get this from an etiology like stroke or so, for instance. It's a result of a very rare genetic disease. And here's the experiment that we did. Let me just tell you a little bit about this because it'll come up again at various points throughout the talk. If you were a participant in the experiment, we show you one of these four faces, one at a time. They look a little strange and grainy for technical reasons. And what we ask you to do is to tell us what the emotion is. So in the simplest experiment, you push two buttons and you say, does this face look afraid or does it look happy? So these two are fear, these two are happy. And if we did this with you with these faces, you would probably be at 100% accuracy in telling me that these two are fear and these two are happy. We don't show you the full face, though. What we do is we show you little parts of a face using a technique called bubbles, invented by these folks down here. And so what you actually see are pictures that look like this. On one trial, you might see parts of an eye, part of a nose, part of a mouth peeking through. And now you have to tell me, is this fear or happy? So this is much more difficult. So randomly, sometimes you see an eye or a nose or a mouth. And for some of these, you can figure out that this is fear or happy, and you get them right. And for some of them, you can't, and you get them wrong. And after many of these, thousands typically, we pay people very well to participate in these experiments. What we can do is we can add together all of these stimuli, all of these little pieces of faces that you got right, where you were able to tell apart fear from happiness. We add all this together, and we subtract all the ones that you got wrong. And what you extract from that is the regions of the face that carry information to tell you what the emotion is. And as you might imagine, if you see parts of an eye or a mouth, you can probably tell if this is fear or if this is happy. If you see the side of the chin, this doesn't help you at all because it looks exactly the same regardless of the emotion. And so this is the information that healthy people would use from these stimuli, from faces, to tell what the emotion is. Doing the same experiment in this patient with amygdala lesions, this is the information that she uses. And these are all statistically thresholded images. We can subtract these two and get this. So what this means is this is the information from faces that your brain uses normally to figure out that a face signals fear or happiness, or fear in particular in this task, that the patient with amygdala lesions is unable to use. So it's an extremely specific impairment. The amygdala is necessary for figuring out information about one particular part of the face, namely the eyes, and that signals fear normally. And if you think about this, there are several stages in processing at which this deficit might arise. So you could imagine in the case of the patient with amygdala lesions that she's looking at these stimuli, little pieces of faces, and doing this task that I just explained to you, exactly the same in the same way that you and I would look at it. So at the level of the retina, she has all the information that you and I would have available, but then her brain doesn't know what to do with that information because she has lesions of the amygdala. That's one possibility. But another possibility is that she doesn't look at these stimuli normally in the first place, and therefore is unable to pick up information about the eye region of the face. And so we can distinguish those possibilities using eye tracking. So if we just show people stimuli like this, so pull pictures of faces, you can superimpose on these how people look at them. So these little white circles are where somebody would be fixating, and the red lines are rapid eye movements called saccades between them. And so you explore the face with your eyes, as I showed you in that little video previously, and you look in particular at the eyes and the mouth. So these are data from a healthy, typical participant. By contrast, the patient with amygdala lesions down here looks at faces like this. So in many cases, she doesn't explore the face with her eyes at all, but just stares at the center of the image. In some cases, she does move her eyes, but it's very disorganized, and it's not preferential for seeking out information from the eyes or the mouth region of the face. So this is summarized here in this slide. So at the top is the experiment from this study, are the results from this study where we show you little pieces of faces and ask what parts of the face do you make use of to judge emotion. And normally it's eyes and to some extent the mouth. And in the case of the amygdala lesion patient, she uses the eyes less. In the bottom, these colored maps indicate by hotter colors where you spend most of your time looking on a face. Normally you look at the eyes. The patient with amygdala lesions doesn't and usually stares at the center. And the reasons I introduced these experiments and show you this graph here is because we did exactly the same study, same stimuli, as I just explained to you, also in a group of high-functioning people with autism. And so if for comparison we have people with autism here, they are in the direction of the amygdala patient. They do use the eyes. They're not as bad as the patient with amygdala lesions, but they use the eyes less than do controls. They compensate by making more use of the mouth region of the face. And again, like the amygdala patient, but not as severely, they look at the eyes less than do the controls. And they look further down generally. So there's some abnormality between the controls and autism, which we can visualize more easily by looking at their difference. So this is the difference between an otherwise matched control group of 10 high-functioning male subjects compared to 10 high-functioning male people with autism. That's what this shows. And the difference here shows that the controls look more at the eyes than use the eyes more than do people with autism, and they look at the eyes more than do people with autism. And so we have some story here about an inability to to pay attention to be motivated to seek out information from the eyes, and to make use of this information to judge facial expressions in autism, and moreover, a similarity between that and the patient with amygdala lesion. So one thing that my lab has been doing is to try and make comparisons across different clinical populations. And by making comparisons, for instance, between a psychiatric population, autism, and a neurological one, patients with amygdala lesions, you could formulate the hypothesis that the amygdala might be one of the structures in the brains of people with autism that explains, in part, their abnormal social behavior. Testing that directly is hard, but we have been testing it in collaboration with Huntington Memorial Hospital here in Pasadena and Cedars-Sinai Medical Center in Los Angeles. And there we collaborate with neurosurgeons. And we have the opportunity to record directly from the brains of surgical patients. So this is at a level of analysis now where we're down here at the level of neural circuits and synapses rather than at the level of large structures like the amygdala as a whole. So let me show you these patients. These are just examples of two patients. These are their, you can probably see this best on these horizontal MR scans. So these are structural MRI scans. The eyes are up here. And these dark horizontal lines here are electrodes that the surgeons have implanted into the brains of these patients, not to do our experiments, but for monitoring epilepsy. So these are all people with epilepsy who, for clinical reasons, have electrodes implanted so that surgeons can localize where seizures originate in order to guide subsequent removal of that tissue. So these electrodes are typically in for about 2 weeks. These patients have seizures while they're in their hospital bed, and the electrodes allow the physicians to figure out where those seizures originate. After that, the electrodes are taken out, and then a neurosurgeon removes that part of the brain where they believe the seizures originate. It's actually a highly successful surgery for the treatment of epilepsy. While they have these electrodes in, they can choose to participate in our research, and we can show them faces and other stimuli while we record from single brain cells, single neurons. And it turns out, very fortuitously for us, that these electrodes, in many cases, are in the amygdala. And so here's the amygdala in the medial temporal lobe, and these little colored dots, which may be difficult to see, are all recording sites from these different patients. So each color is a different patient, and you can see the recording sites in the amygdala in each of them. So once the electrodes are implanted, they get structural MRI scans with the electrodes in their brains, so we can see exactly where the tip of the electrode is located and where we are recording neurons. So coming back to the picture that I showed you previously, we had this finding that if you show little pieces of faces to people, it turns out they make a lot of use about the eye information, about the eye region normally. The patient with amygdala lesions fails to do that, and people with autism make less use of the eyes, but seem to make exaggerated use of the mouth. And so we did exactly that same experiment also in these neurosurgical patients. We showed them these little pieces of faces while they're pushing buttons to tell us if this is fear or is this happy. We replicated the same finding that I just showed you, so this is just what we showed you before. So whereas controls make a lot of use of eye region and to some extent the mouth, people with autism use the eyes much less, but also use the mouth. This is much noisier looking because we only have 2 patients with autism. But what we had here were 2 patients who had electrodes in their brains for the treatment of epilepsy, who also happened to have a diagnosis of autism. That's this group here. And then we had 8 controls who are people with epilepsy who have electrodes implanted in the amygdala, but do not have a diagnosis for autism. You might ask yourself, how did this come about? It turns out that autism and epilepsy often go together. And so by chance, every once in a while, we would have a surgical patient with electrodes in the amygdala who also had a diagnosis with autism. What we can then do is record from these neurons in the amygdala. So all the recording sites from the epilepsy patients without autism are in blue, the ones with autism in red. They're both in the amygdala. And we record waveforms like this from single cells, so potentials, electrical potentials of neurons in their brains. And now we can ask not the extent to which people are pushing buttons to tell apart these emotions, but we can ask which parts of the face, if we show an eye or a nose or a mouth, are driving neurons in the amygdala. And what you get from that is a picture that looks like this, which is very similar in many respects to the behavioral data. So here the hot areas show the regions of the face that are most potent in making neurons in the amygdala fire. So in the epilepsy subjects without autism on the left, it's the eyes and to some extent parts of the mouth that, when shown in these little pieces, are most powerful in driving amygdala neurons. Whereas in the autism subjects, the eyes are not driving these neurons and only the mouth is. So we find here a correlate at the level of single cells in the amygdala in people with autism that may explain in part their behavior and their propensity to pay attention to the eyes or the mouth abnormally when they look at faces. Now, everything that I've told you about, well, on the one hand, it allows us to really drill down and try and get a handle on the mechanisms whereby people figure out social information from looking at parts of the face and so forth. But this is very artificial if you think about it. People with autism certainly don't come in complaining of an inability to look at the eyes in photographs of faces. They complain of actually interacting with people normally in the real world. So one thing that we and many other labs have been at pains to do is to extend these findings in the lab, where typically you show pictures of people or stimuli on a computer monitor and so forth, to something that's more ecologically valid and translates to the real world. So the idea is, can we go from the lab out to closer to this level of social functioning, which is ultimately so relevant? And we've been doing that, so to some extent you can do this, even in the lab. So here's an example using eye tracking, where we're asking not the question of how you look just at a photograph of a face, but how you look at a real person, still on a screen. But this is a live person who's interacting with you through a live video conference, and you've just met this person, they're in the adjacent room, you're talking to them, the face is moving. It still allows us to quantify things because the face is nicely on a screen and so forth, but it's a real person that you're interacting with in this live video conference. We can go further than that. It's hard to quantify, or a lot of work to quantify, but we can actually measure how you look at and interact with a real person who's sitting across from you. As you might imagine, it's difficult to control for everything here. In particular, you want this person who's the stimulus in this experiment to be as reliable and replicable across subjects as a photograph of a face would be. And so this guy used to be on The Sopranos as an actor. He's a professional actor, and his character got killed early on in the show. And so we hired him, and we videotaped him and checked, and indeed he was extremely reliable. So he's always looking at the subject with a neutral facial expression, maintaining direct eye contact, and going through a very scripted set of questions and answers. So to some extent, this is still not like real life. It's somewhat artificial, but it is a real person that you're interacting with, which sure feels different than just looking at a photograph. So the kinds of data that you would get look like this. So this is taken from the scene camera. There's a little camera on the eye tracker on the forehead of the research subject. So this is the view of the research participant looking at this actor who's asking various questions. And the green circle that's hopping around there is the direction of gaze of the subject. So they're moving around and talking, which is why the actor's head keeps bopping around. and you can tell that they're looking in various places, including at the face and the eyes of the actor. So we go through frame by frame of this video, very labor intensive, and use a semi-automated face detection algorithm to pull out the face from every frame, align those, and then superimpose where the subject was looking. And so both in the live video condition and also in this face-to-face interaction, healthy, typically developed participants spend a lot of time looking at the eyes. So again, these color plots here, the hotter the color, the more time the subject spent looking there. So as was the case with the photographs, you also look a lot, at least in this particular kind of scripted social interaction, you also look a lot at the eyes of this actor. We did the same experiment then with our patient who had bilateral amygdala lesions. And what we found there is shown in the bottom here. So she failed to look at the eyes and instead kept looking at the mouth of the actor. And there's various explanations that you can come up for with this, that the mouth is moving, at least on some occasions. It's sort of the next most salient region compared to the eyes. But this was highly reliable and is a pattern that we're now also starting to find in high-functioning people with autism. Those data are more complicated, so I'm not showing them here. But it gives you a flavor of the direction in which we're going. As you can imagine, this quickly sort of blows up in your face if you start adding more and more complexity. So you might quickly ask, well, what if this actor isn't always looking straight at you? What if he's smiling? What if he's a familiar person, or male or female? Well, I'm sure all of those will make a difference. And so trying to account for that contextual complexity is a huge challenge. So we're trying to extend this out to more ecologically valid kinds of stimuli, but it's hard because the world's really complex and unconstrained. Let me show you another example, though, now with people with autism. This is work by my former postdoc, Dan Kennedy, who's now faculty at the University of Indiana. And again, he was very interested in the criteria that are used to diagnose autism and processing complex social stimuli and enjoying watching social stimuli. And there's a lot of examples of that around. So what he used as a stimulus were episodes from The Office, with which many of you are probably familiar. And if you watch this, almost every frame of The Office has faces. It's full of people, complex social interactions, and of course it's designed by the people who produce this to be maximally attention-grabbing, rewarding, and socially engaging. That's why people watch it. So in a sense, the producers of The Office have sort of created for us an ideal social stimulus. Healthy people watching this really get drawn into it, and they care about the characters. They make complex social inferences about what people are doing. They look at the faces. They recognize the emotions behind the face. And so we thought, well, let's use this as a stimulus. It's much more valid, and people really enjoy looking at watching it than if you just had just a picture of a face statically up on the screen. But it's still replicable and reliable, and you can show the same video over and over again to people. And so in that sense, you have much more experimental control than you do using a live person as a stimulus. And so you would show people this. It goes on for about 20 minutes, and there were other various control videos we showed as well. And people really get into this. So they really are very engaged. We can ask them questions about what they remember afterwards. We can get ratings of this movie at many different stages. So we have this stimulus extremely well quantified. And we can ask, at what point, for instance, is there a face on the screen? At what point is there a socially awkward moment? At what point is an emotion expressed? And so we have all of that, and all of that unfolds in this dynamic video. And we can then use eye tracking while people are watching this video. So while you're watching this video, if you look at the red cross, this would be where a subject is looking while they're watching this social stimulus. There's other ways of depicting this. So you could do it with this sort of heat map here. So here, this indicates where a subject would be looking, where the direction of gaze is, as the movie scrolls by. Now we take a group of subjects and do exactly the same thing, and you would have something like this. So what this would mean is that most people, say you have a group of 10 subjects, 7 of them would all be looking here, 1 subject is looking over there, maybe 2 are looking over here, and so forth. So in a group of subjects, you would get regions where most people are looking and some regions where a few others are looking. And again, you would have this unfold over time throughout the whole movie. And you can see that at certain points in the movie, everyone looks at one spot. They all look at one place, and then at other points of the movie, there's more spatial dispersion, and they look in various places. So just to walk you through how you would analyze this, it's fairly straightforward. You would go through frame by frame, and you can then quantify how typical a given single subject's eye tracking is to the stimulus. So you can quantify the similarity to the whole group over time. So for instance, if you have this frame up here and the subject looks there, that's a location where none of the other subjects in the group looked, and therefore their similarity would be around 0. In the next frame, they look over here. So a few subjects, but not the majority, also looked there, and so their similarity would be something like this. Over here, they look where the most people looked, and so their similarity would be the highest. And so you do that frame by frame through the whole movie, and you would have a plot like this that shows you at various points in time the similarity to how typical, how standard a person's viewing is over time. There's some places where this person looks where everybody else looks, and some places where this person would look where very few or none of the other people look. So the study that we did was with these two groups, controls and autism, that were otherwise fairly matched on IQ and so forth. And what we found was, these are the curves, now the similarity in viewing patterns for the controls in blue, autism in red. What we found was that by and large, the autism group also looked where the controls looked, with a few exceptions at a few points in time. And so if you just quantify that over the whole movie, shown on the right here, this is the range of the controls in blue and autism in red. And you'll see that on average, the autism subjects did look abnormally at this video. A few individuals very much so, and many of them were fully in the normal range. So it's a small effect. It's an effect that you can only see at the group level. But the really interesting question then is, well, what happens at these points here, particular points in the movie, where there is a statistically significant difference between where the autism subject and the controls look. So if you look at a longer time frame, you can see it more. There's many specific points in time where the red and the blue curves are different. So these are places where the control group and the autism group do look differently at the movie, and we can ask what's happening at that point in time. So this is something that we're still working on, but one thing that we can quantify is just the low-level visual saliency of this video. So regions where there's strong color or contrast, changes in orientation or flicker, that would sort of automatically guide your visual system to pay attention to those regions. So these hot colors here are the result of a computational model. These are not data from subjects yet, that would tell you where these regions, where there's high contrast, etc. And what we found is that the autism group at these particular points in time, and on average across the movie, where they look tends to be driven more by these low-level visual cues, just if there's something bright there or if there's an edge there, whereas in the controls, they are driven less by those simple visual cues and presumably more by the semantic social meaning of what it is that they're watching. And so we can do lots of things with this. So if you look at the similarity to the control viewing pattern, how abnormal on the right, on the left, or typical on the right, your viewing pattern is to this video, there's a correlation, not a great one, but nonetheless a correlation between how well you understand what's happening socially in the movie. And again, the red dots here are autism individuals, the blue are controls. We have a correlation also between a score that quantifies autism symptomatology through a questionnaire. So this is independent of the movie. This is just autism symptomatology in the real world. And again, we find a correlation with abnormal viewing pattern on this movie. So there seems to be something that these eye tracking patterns are giving us from this movie that taps into autism symptomatology, that taps into the ability to understand, to extract and understand the social meaning of the stimulus. And so this is a whole direction of work that we're still analyzing and going forward with. I should add that all of this was also done inside an MRI scanner, and so we actually have 20 minutes of brain activation patterns that we're correlating with these eye movements as well. Huge amount of work still ongoing, none of it published yet.